Good morning, students. Today I am going to synchronize pharmacognosy with pharmacology. As a part of this lecture, here I am taking physostigma. Physostigma is an indole alkaloid. So, the examples of indole alkaloids includes ergot, nux vomica, Raulfia and Vinca. So these are indole alkaloids. Indole alkaloids are obtained from, these are all indole alkaloids. Indole alkaloids are obtained from tryptophan. Generally, alkaloids are obtained from amino acids. Here indole alkaloids are obtained from tryptophan. So coming to the physostigma. Physostigma consists of dried seeds of Physostigma venenosum. Physostigma venenosum. Family is Leguminosae. Family Leguminosae. So physostigma, the main chemical constant in physostigma is physostigmine. Physostigmine is the main chemical constant. It is also called as aceriin. So aceriin, when is exposed to air and light, it gives rubaceriin. Rubaceriin is a red color compound obtained from the aceriin. When exposed to air and light, the aceriin undergoes oxidation and it produces a red color compound, that is rubaceriin. So this is about pharmacognosy part. Next, coming to the pharmacology part of physostigmine. So here it is a reversible anticholinesterase, physostigmine or aceriin. It is a reversible anticholinesterase or indirectly acting cholinergic drug. A reversible anticholinesterase. Reversible anticholinesterase, it is also called as indirectly acting cholinergic drug. So after inhibition of the cholinesterase enzyme, so increase the acetylcholine level. This acetylcholine bind to the muscarinic receptors. Muscarinic receptors are cholinergic receptors and produces cholinergic actions. So the main therapeutic benefit with the physostigmine is it is mainly used in the treatment of glaucoma. Because it decreases intraocular pressure. Physostigmine decreases intraocular pressure. Being a tertiary amine, it is highly lipid soluble drug. It enters into the central nervous system and easily crosses corneal layer. Because of this reason, it is used in the treatment of glaucoma. And physostigmine also used in the treatment of atropine poisoning. Overdose of atropine produces toxic effects like tachycardia, blurred vision, constipation. These problems minimized by physostigmine. Not only atropine poisoning, physostigmine also used in the treatment of tricyclic antidepressant poisoning. Tricyclic antidepressant poisoning. So because tricyclic antidepressants also having anticholinergic actions, these actions are minimized by physostigmine. So this is the advantage of physostigmine. So pharmacologically it has several therapeutic uses. So chemically it is a carbamate. It has two chiral centers. to chiral center. So this is about physostigmine. And the next indole alkaloid is nux vomica. So nux vomica, so the biological source is, it is consists of dried seeds of Strychnus nux vomica. The family is Loganiaceae. Family Loganiaceae, nux vomica. So main chemical constants of nux vomica is strychnine and brucin. Strychnine, brucin, loganin, alpha caliburin, vomicin. So these are the main chemical constants of nux vomica. Here strychnine. Strychnine induces convulsions. So why strychnine produces convulsions? Here strychnine antagonizes the action of glycine. This is a glycine antagonist. So glycine basically it is a inhibitory neurotransmitter. So this inhibitory neurotransmitter action inhibited by strychnine and produces excitatory action. This leads to convulsions. So strychnine is act like a CNS stimulant because it antagonizes the action of inhibitory neurotransmitter, that is glycine, and produces convulsions. Next, brucin is for dog poisoning. Brucin is for dog poisoning. So this is about chemical constants of nux vomica. So another important feature of nux vomica is that is it has lignified trichomes. Lignified trichomes present in the nux vomica. This question has been asked several times in competitive examinations. Which alkaloids having lignified trichomes, that is nothing but nux vomica. So this is about nux vomica. So next drug is Rauwolfia. So Rauwolfia also indole alkaloid. Rauwolfia, synonyms of Rauwolfia are Sarpagandha and Chota Chand. Sarpagandha and Chota Chand. Coming to the biological source of Rauwolfia, consists of dried rhizomes and roots of Rauwolfia serpentina. Rauwolfia serpentina, family Apocynaceae family. So coming to the microscopic characters of Rauwolfia, here the main important feature is that is microscopic characters. The first one is stratified cork. Stratified cork is present in the, it divided into 2 to 8 zones. Stratified cork is the major feature of Rauwolfia. And the second one is sclerenchyma and stone cells are absent. Stone cells are absent. This is also another feature. So when we are going to compare with the Rauwolfia, another species, that is Rauwolfia tetraphylla, this is another species. So this Rauwolfia tetraphylla non-stratified cork cells. Non-stratified cork and stone cells are present. So this is the difference between Rauwolfia tetraphylla and Rauwolfia serpentina. It is an indole alkaloid. It is indole alkaloid obtained from the tryptophan. Coming to the microscopic coming to the chemical constants of Raoulfia. So here chemical constants includes reserpine, resinamide and ajmalicin. So reserpine is the major chemical constant. It has very therapeutic importance. Reserpine on hydrolysis. So hydrolytic pathway is very important. Here reserpine on hydrolysis, it gives reserpic acid plus methyl alcohol and trimethoxy benzoic acid. So reserpine on hydrolysis gives reserpic acid, methyl alcohol and trimethoxy benzoic acid. When coming to the resinamide, so resinamide on hydrolysis, it gives same products similar to that of reserpine, but instead of trimethoxy benzoic acid, it produces trimethoxy-cinnamic acid. Trimethoxy-cinnamic acid. Same, it produces reserpic acid and methyl alcohol. So compared to resinamide, reserpine has therapeutic benefit. And coming to the ajmalin, so ajmalin is basically the function is similar to that of quinidine, like it produces quinidine-like effect produced by ajmalin. So that's why it is used in the treatment of arrhythmia, cardiac arrhythmia. This is very important. Ajmalicin for cardiac arrhythmia. So coming to the pharmacology part of Raoulfia, so here the major chemical constant is reserpine. So here reserpine, it is a catecholamine depleter. Reserpine is a catecholamine depleter. So it involved in the degradation of noradrenaline and dopamine. So 5-HT also degraded because of this reserpine. Reserpine inhibit the entry of noradrenaline and dopamine into the synaptic vesicle by inhibiting a transporter known as vesicular monoamino transporter, that is VMAT. So generally the neurotransmitters enter into the synaptic vesicle with the help of VMAT, noradrenaline or dopamine or 5-HT. So this action is inhibited by reserpine. It inhibits the VMAT transporter. So there is no entry of noradrenaline and dopamine into the 5-HT into the synaptic vesicle and increase the concentration in the axonal plasm. This noradrenaline, dopamine, 5-HT get degraded in presence of monoamino oxidase and decreases activity of these monoamines. Okay, so that's why it produces effects like hypotension. If noradrenaline level decreases, it produces hypotension. So if dopamine level decreases, so it produces Parkinson-like symptoms. And if 5-HT level decrease the activity of 5-HT leads to increase the appetite, produces weight gain. Because of these reasons, reserpine is withdrawn from the market as a drug. So this is about Reserpine withdrawn from the market because of it produces hypotension, because it is involved in the degradation of noradrenaline. So, and dopamine also get decreased because of reserpine, produces Parkinson disease. So 5-HT activity also decreases. This leads to weight gain. So weight gain. So this is about reserpine. Next coming to the vinca. Vinca also indole alkaloid. Vinca is obtained from the entire plant of Catharanthus roseus. Catharanthus roseus, again family Apocynaceae. So I will discuss only few important points about vinca, which is helpful for the competitive examination. So coming to the here microscopic characters, here the important feature is that is it contains cruciferous stomata. Cruciferous stomata. That is nothing but the another name of cruciferous stomata is anisocytic stomata. Generally, this type of stomata is present in the Solanaceae family, like Atropa belladonna, Datura. This kind of stomata present in the Solanaceae family. But here vinca also has cruciferous stomata. It is nothing but anisocytic stomata. So vinca main chemical constants are vincristine, vinblastine and vindolin. Vincristine, vinblastine, vindolin. These mainly vincristine and vinblastine is used as an anti-cancer agent. Anti-cancer agent vincristine and vinblastine. So vinblastine mechanism of action is it inhibit the microtubule formation. So after inhibition of this microtubule formation, generally these two drugs bind to the tubulin protein and inhibit the microtubule formation and further inhibit the DNA synthesis. So generally used to Ewing's sarcoma. This is drug of choice for Ewing's sarcoma. It is a soft tissue cancer, Ewing's sarcoma, bone or soft tissue cancer. Ewing's sarcoma is drug of choice for Ewing's sarcoma. And next coming to the adverse effects. So the adverse effects are similar to that of all anti-cancer agent, but mainly it produces, that is neuropathy. Neuropathy is the major drawback of vincristine and vinblastine. So this is about Catharanthus roseus or vinca. It is also a indole alkaloid. So mechanism of action is very important. That is microtubule, inhibit the microtubule formation and inhibit the further DNA synthesis, and cruciferous stomata also present in the vinca. Here we are using entire plant. Okay, so this is about indole alkaloids, pharmacognosy and pharmacological actions. Thank you.